So in the first part of my talk, I'm going to discuss the duality between robustness and evolutionary decay. And the argument is that as systems acquire mechanisms that protect them from external perturbations, those mechanisms which protect the system from perturbations also shield internal components of the system from direct environmental challenge. So systems acquire mechanisms that protect them, shield the internal components. The internal components are not exposed as directly to the external challenge and will tend to decay over evolutionary time. So there's a natural duality between robustness and decay. And I'll suggest that this duality between robustness and decay influences the evolutionary dynamics of the design of biological architectures and biological computations. So that's the first part of the talk. In the second part of the talk, I'm going to look at general aspects of input-output relations in biology. You can think of inputs as sensors or measurements of extrinsic information, and the outputs as the computational transformations of those inputs. And one interesting pattern that often occurs in input-output relations in biology is that we see switch-like behavior. That is to say, when the inputs are low, the outputs stay low, and then over a very narrow range of inputs, the outputs switch to a high level. So we're getting a switch, which is basically an analog to digital converter and plays an important role in building components of computational architectures in biology. And the puzzle with these switches is how they arise from biochemistry, because the basic laws of mass action chemistry don't look like that. Basic laws of mass action chemistry give us an input-output relation that is not at all switch-like. And so the puzzle that recurs is the mechanisms by which biology builds such analog to digital switches. And one reason for focusing on this is that it forms a nice little model for understanding the little pieces that build circuits for biological computations. And one of the arguments I want to make is that in order to understand these computations, that you need to pay attention to the aggregate structure of input-output relations. That is to say, inputs come in, and they're transformed in multiple ways before you get outputs. And so if you look at the aggregate structure of the input-output relation, you can understand things a bit more deeply, which is just to say that aggregation matters, which is a very widespread and well-understood point. And the second key issue is that we need to look at scale transformation. So we're led to the classic aspects of aggregation and scale in understanding these components. I think this will help us to understand the design of biological systems. So the first part's about the duality between robustness and decay, and the second part about input-output relations. And although those may seem like very different topics, towards the end I'll suggest that they're actually closely related through this notion of understanding aggregate structure. So that's the talk. So let's go to the first part of the talk, which is the duality between robustness and decay. And let me introduce this by giving you a little story about the history of computer hard drives. Very crude story, but I think it'll capture the essence of my main point. If you're old enough to remember 30 years ago, you'll remember that when you bought a computer hard drive, it hurt. It cost a lot of money, and typical mid-range computer systems that were used in companies often stored data on single hard drives that were very expensive, and the goal was to engineer them to high tolerance and low error rate. But over the last 30 years, there's been a real change in the architecture of data storage in sort of mid-range computer systems. Now almost all mid-range computer systems that companies use redundant arrays of independent disks, or RAID arrays. And what a RAID array does is when the data come in to be stored, is broken up into little chunks and spread across a series of disks in a form of redundancy. But it's not just simply making copies like backup copies. It's more complicated than that. And the data are stored sort of across these different disks. And the locus of design has shifted. Instead of engineering the individual hard drives to high tolerance and low error, instead there's a board of logic that sits above the system and controls the system. So the data come in, and this board controls spreading the data out and recovering the data. So really what we've had is a shift in the locus of design from the focus on engineering the individual disks to high tolerance to engineering the system to high tolerance with the focus on the controller board. And associated with that, and this is the interesting point, is that in effect what's happened is that people and companies now use cheaper, lower performing, sloppier hard drives. It doesn't pay to have a very expensive hard drive, because in these redundant systems, if a hard drive fails, you just pull it out and stick another drive in. And so companies tend to use sort of modestly performing, relatively cheap drives that are not engineered to the tolerance that could be achieved. And I think this process of the migration of design, locus of design and control to a higher level to achieve better system performance associated with a decay in the performance of the individual components is something that inevitably happens in the history of design. I think it's been very important in biology. So you can sort of think the logic is very simple, and probably you can see the logic. Now, what I would like to be able to do is give you a whole series of examples showing you how you can understand the history of evolutionary design, computational architectures in biology based on this argument. But in fact, I don't have a good example. So I'm in a relatively weak position. But nonetheless, I think it's very important. And so what I want to do is give you a couple of predictions about how I think we can begin to see how this idea might work. I should also say that one interesting consequence of the idea is that there's an irreversibility. Once you get a higher level controller and you get a decay in the underlying components, you can't take away the higher level controller because the components no longer perform sufficiently for the system to survive or to function properly. So there's an irreversibility in the history of evolutionary design in this way.
I mean, prokaryotic to eukaryotic gene regulation
Great.
sounds like an example, but
it's not. So we'll get back to it. So the example that I want to give you, which I think, and this is just a hypothesis, but one of the things that's come up with high throughput genomics and the ability to measure lots of things, until a few years ago, the measurements were always done in aggregates. You couldn't measure single cells. The technology wasn't there. But that's changed in the last couple of years, and people can now measure the protein levels and the messenger RNA levels in single cells. And one of the discoveries, which some people think is surprising, I don't, is that there's a high level of stochasticity from cell to cell if you look at the protein expression levels. And so thinking about that, this is the way I would think about it in terms of what I just said. If you imagine a tissue composed of many cells, and you think of the function of the tissue as being to secrete a hormone or an enzyme that works at a distance, right? Then what matters? What matters is the average secretion rate per cell. And the variability among the cells doesn't matter at all. Put that another way, the best way to achieve high precision is to aggregate a lot of components because the variance of the average is very small. Once you do that, you achieve a high level of robustness in some sense. You can use a lot of sloppy components. You have a lot of precision, but the selection in each, the regulatory design in terms of the tolerance of each cell have really created a situation you can have very poor tolerances per cell. You expect a high level of stochasticity at the cellular level. Contrast that with a situation, for example, let's say in bacteria. Bacteria often secrete enzymes to digest resources in the local neighborhood outside of the cell. Bacteria might be secreting enzymes to digest these resources, and if we think of a viscous environment where the diffusion rate is low, cell density is low, then what a single cell does matters for its ability to acquire resources. So the functional level is actually at the single cell level, and so we expect the regulatory system to be designed to much higher tolerance and lower sloppiness. So we get a simple prediction based on the level of function. We need to pay attention to the level of function. We get predictions about the degree of sloppiness. This is a very simple way to make a prediction about the observations that are coming in about the level of stochasticity that's observed in different systems. I wouldn't be surprised if somebody else has said a variety of things like that in the literature. The literature is big. Pretty much everything gets said. It's not a claim of originality, but it's not the dominant view in the literature. And I think this gives us some way of looking at these problems. So that's in essence the first part of the talk. And I think that the main point is that if we want to look at the evolutionary history of the computational architectures as they evolve and are designed by biological processes, we need to understand the sort of weird feedback that happens with robustness or insensitivity or the migration to higher levels of control and the decay of the underlying components. And I think that's going to give a real directionality to evolutionary history. So the main arguments here are in a couple of papers or some others. Maladaptation and the paradox of robustness and the evolution of robustness and cellular stochasticity. Now there's a huge literature in biology about robustness. Most of it focuses on genetic variability. Doesn't focus, the key to my argument is that anything that makes the system insensitive to perturbations changes the internal marginal costs and benefits and the design of the components that were there previously. And I think that that's the, I don't want to say novel, because I'm sure you can all find some precursor to my idea, but not what's often discussed in the literature, although there's a huge literature on robustness. So that's the first part of the talk. We'll come back to that at the very end. Now I want to turn to the second part, which is this analysis of input-output functions in biology. And I've told you that we often have switch-like behaviors that we observe, and that the key puzzle is that that contrasts with basic laws of mass action chemistry. So here's basic mass action chemistry, Michaelis-Menten kinetics, which is sort of the foundation of any biochemical analysis. And this is the sort of pattern you would expect for an input-output relation based on Michaelis-Menten kinetics. Here you imagine some input signal coming in at concentration S, and imagine that the cells have some non-active molecule that's around in some concentration. And what S does is transforms the inactive molecule to an active state. Right? So the greater S, the greater the concentration of the active molecule. So that's all. The signal's driving the activation of a particular molecule. Michaelis-Menten kinetics for a classic reaction would tell us that the input-output relation would be a diminishing return. And that's sort of the basis that we use for doing any biochemical analysis. But you can see biologically that that's already a little bit weird, because think about it. That says that you're getting maximum sensitivity and infinitesimal concentration of an input, which, if you know anything about biology or you think about biology, just that doesn't sound right. You're not going to be getting maximum sensitivity at infinitesimal concentrations. And in fact, many biological input-output relations look like that. They're more switch-like, analog to digital. And this is so common in input-output relation that it has a name. It's the Hill equation pattern. And the Hill equation pattern is discussed in the history of biological literature over the last 100 years. There's a vast literature on this. And one of the interesting things is that this very simple S-shaped pattern arises at many different scales, from biochemical reactions done in isolation in the laboratory to input-output relations in cellular systems and regulatory controls in systems biology. Systems biologists frequently invoke the Hill equation pattern in order to build their little circuits in their models. And it's the fundamental equation that's used to fit dose-response curves at the whole organism level. That is to say, if you take a whole animal and you treat it with a concentration of a drug, how does the animal respond to the drug? That's in pharmacology, a dose-response curve. The fundamental method for analyzing dose-response curves is to fit a Hill equation to it. The Hill equation is incredibly simple. If we do a non-dimensional form, an input x and an output y, we have one parameter k, which gives us the different curves that are fit. If k equals 1, we have Michaelis-Menten classical mass action chemistry. As k increases, you get a more switch-like behavior. But the remarkable thing is that this incredibly simple equation actually gives a fairly remarkable fit across a huge range of scales in biology. So most of what I'm going to talk about is from this article, Input-Output Relations in Biological Systems, Measurement Information in the Hill Equation. So in biology, this switch-like behavior is often called bistability, because the system is stably in an off state until the signal gets strong enough and drives it to a stable on state. So the question is, how does the signal drive the system from one stable state to another? So the question is bistability. It's often discussed also as ultrasensitivity, because the system is ultrasensitive to changes in a narrow range. So this is very useful in terms of components and architecture for computational systems in biology, and it's something that's seen quite frequently. And so people wonder about, where does it come from? And so here's just one example of a study. The literature on this is vast, but here's one study which I think is well done. And the question here is the regulatory control of mitosis. Mitosis is cell division. You can imagine that if a cell, for controlling cell division, you either want a cell to be in a non-dividing state or going through division. It doesn't pay to be kind of dividing. And so you need some sort of analog to digital converter. And in particular, in this case, CDK1 is one of the regulatory molecules which is important in driving the cell into a mitotic state. CDK1 modifies this particular molecule, and so it's this molecule which is actually, in this particular case, used as a proxy for determining the degree to which CDK1 is driving the cell into the mitotic state. And you get a nice Hill equation pattern with a Hill coefficient of about 3.5. Now in this article, the authors posed the puzzle exactly as I posed it. Classic Michaelis-Menten kinetics predicts this, but what we see is this. So the question, and here what I'm really going to emphasize, especially for people in computer science, is the mindset. You've got a pattern, you've got to find the molecular generative mechanism. That's the goal of molecular biology. How do you generate that pattern based on molecular process? So what they say is several generative mechanisms can in principle give rise to ultrasensitivity, and then here's a list of 3 classic generative mechanisms. And so you have a shopping list of generative mechanisms, microscale mechanisms that generate the pattern, and you want to figure out which of those is operating in this case. So you do a study and you figure it out, and there
I
can...
just asked, I mean, the very simple one of just everything forming dimers, or trimers,
There's lots of them.
that wasn't on your list.
I have a longer list coming up.
Oh,
Hang on.
sorry.
Relax. So the mitotic regulators, so this is the conclusion from their article, the mitotic regulators can inactivate each other through inhibitory chemical modifications, phosphorylations, and then this is their main conclusion. There's a double negative feedback loop that forms a bistable trigger that makes the transition into mitosis digital. Okay? So that's the conclusion from their article. Now that's fine, I don't have any real criticisms of that. In fact, they're probably correct, because it's a very good lab, and I'm sure the study was done very well. But I have a little reservation deep inside of me which says, when you get a pattern that's that simple, that matches an equation which has so simple across every scale in biology that recurs over and over and over again, maybe always fitting little microscale processes to the pattern isn't the right way to think about the problem. In fact, what I think is, I see that. Okay? That is a Gaussian or normal distribution. No biologist would see a pattern from a study that was a Gaussian pattern and try to come up with a specific generative mechanism to explain that pattern. They all know that this arises by the aggregation of a bunch of sloppy things. You throw a bunch of things together, you add them up, and you get that. Somehow that was trained, everybody kind of gets that. Any other pattern in biology deserves a very special microscale explanation. Now, it's not true that the Gaussian is the only attractor in the world. There are others. We'll get back to that issue. Say it.
Yeah, so I was wondering, if you take the example of hemoglobin, that's actually when the Hill equation was developed. And if you look at the history of the studies in hemoglobin, you see that with more data, it became clear that the Hill equation is not a good model. In other words, you eventually...
I know the history of the Hill equation, and I'm
Right,
not
so-
talking about that because that's irrelevant for what I want to say.
No, what I mean is that
I mean,
eventually...
just because it's true doesn't mean it's relevant.
No, what I mean is that eventually you got to the point where the molecular structure makes conversions between 2 states, right?
I know all of that. It's
So-
not
so-
relevant for what I want to say.
Okay.
If you want to talk about the molecular biology of hemoglobin, we can have a discussion about the molecular biology of hemoglobin. But it's not the key point. I understand the distinction between specific explanations for specific molecules and the history of how people have understood the Hill equation. But my main point is that in spite of the fact that some people understand hemoglobin, that over and over and over again, when people see the Hill equation pattern in molecular biology, and I'll give you many examples in a minute, that people have tried to make microscale generative explanations rather than thinking about the problem as potentially an attractor. And that's because biologists don't recognize any attractor except the Gaussian. That's it. Of course, that's not 100% true because lots of people understand the power of aggregation. The prize for the best title goes to Stuart Kauffman, The Origins of Order. Order is pattern. The Origins of Order to Kauffman, he understood that aggregation was very powerful in creating pattern, and that's what his book is about. And so I think he really understood this fairly deeply for molecular biology. He had the right idea. If you've ever tried to read the book and you've gotten anything out of it, I'd like to talk to you. It's full of good ideas, but so it's a very chaotic book. But I think we have to give him credit because he did understand very deeply this issue. And this really is the heart of the book, in spite of the fact that he tried to explain everything else on top of it. And so that was the problem. And there's other people. Nick Barton has over the last several years, developed a program with considerable success in evolutionary genetics and looking at the powerful role of statistical physics. And I think this is an ongoing project which is continuing to develop, and there's a lot of success here. In ecology, there's a long history of looking at aggregation. Yohei Wasa and Simon Levin 25 years ago wrote some nice articles, and this has developed into a subfield of ecology. So it's not that I'm the first person to think that statistical physics might be interesting, but in spite of these beautiful developments and beautiful articles, and this is really good work, this is a very tiny minority in the way biologists think about problems. They do not think this way, generally. Instead, you get distinction, you get a pattern, you come up with a microscale explanation to fit the particular pattern, and in fact, you might be correct in any particular case. So I'm not going to argue with the particular cases, but I get suspicious because this pattern is so simple about whether always they're getting the right answer. I continue to see this. I see attractors, I see aggregation as being often the best explanation for this sort of thing. Now let me try to convince you a little bit.
So by attractor, like, you know, my guess would be for the threshold model, the attractor involves selection, whereas for the Gaussian, it's an attractor that's operating by processes that don't include selection. Is that a distinction that matters here?
Doesn't matter to me, but
Yeah, but I mean, I think that may explain it. That if it required selection, then you don't call it an attractor anymore. I think that might be part of the biology mentality.
see what you think after I give you my view. I'm still warming up. That's okay, but I haven't given you some ways of thinking about it, alternative ways of thinking about it. I'm still trying to convince you here because I'm making strong claims about what other people think, which is always a dangerous position to be in, because of course those people are never in the room, you're kind of making somebody up. But I want to try to convince you that it's actually, there's some truth to this claim, because it's important for what I'm trying to say. This is a review article that came out 2013, Ultrasensitive Hill Equation Response Motifs, Basic Amplifiers and Molecular Signaling Networks. This is a beautiful review article. I truly think it's very well done. These people are from the Center for Dose Response Modeling. The reason why they're from the Center for Dose Response Modeling and they're writing an article about molecular biology at the detailed molecular level is because dose response modeling is based on the Hill equation. These people are experts in the Hill equation, and they're using their expertise at the molecular level to explain these patterns. So here's one part from just one example from their article. You know, you've got an active form and inactive form. This is a classic explanation from the history of biochemistry. You've got an input that drives the system from inactive to active, and you've got a repressor that drives the system from active to inactive. If the forward and back reactions are near saturation, then you get a very strong switch-like behavior biochemically. And this has been known for a long time, and this is one of the classic ones on the shopping list of potential molecular explanations. It works. It's well known, requires forward and back saturation. Here's another nice idea from the article. The key about the Hill equation pattern is that you get low sensitivity at low input before you get a rapid rise. So what's a simple way to build that molecularly? You can titrate out the input signal when it's low. So here's the input signal. D is a receptor that binds the input signal with high affinity. So as L comes in, it binds to D. D doesn't do anything, just titrates out the input signal, so you don't get a response. When D is saturated, then L starts to bind to R, which is a receptor, which then transduces a signal. So you can build the Hill equation response very nicely by just titrating things out. It works. I have no doubt that this exists in biology because pretty much everything that can happen does happen. And here we go. I'm sure, maybe your idea's in here, I don't know. But this is from their article, all the different alternative motifs that they've reviewed. And they're not claiming this is all of them. They're just saying here are some ones that have been discussed extensively. Here are the two I gave you. Here's a bunch of others. You can build a Hill equation response from biochemistry in a large variety of ways. The literature on this, both in the laboratory and in theory, is immense. They're microscale explanations to fit the pattern. To summarize, in this same review article, here's a particular reaction, a known biochemical reaction from real biology. Here's a Hill equation coefficient, and here is which of the ones on the shopping list is thought to match that pattern. Okay, so this is the state of the art, is you just take every example and you match it, figure out the biochemical mechanism, and there's a lot of them. Okay, so biologists do think this way. And they could be right in each case, but I don't have an argument with any case, but I have a profound suspicion. It is so simple, that equation is so simple and fits the patterns so well, that is it really right always to be thinking in terms of specific, detailed, deterministic, microscale explanations to fit those patterns? I wonder. I really do wonder, and keep on seeing this in my head. Now everybody knows the central limit theorem, but I personally never get tired of looking at these curves, because to me this is just amazing. Okay? So what I did, this is just a little example. These are probability distribution functions at the top. So you have some x value. The height is the relative probability. We're going to sample from these distributions 2, 4, 8, and 16 times and add them up. By the time you go from here down to 16, or from this distribution down to 16, that Gaussian shape is perfect. That pattern is perfect. That's because the Gaussian is the outcome of the dissipation and loss of information. The only thing that's retained is, roughly speaking, the average deviation from a central location. Okay? The dissipation of information leads to perfect pattern by the loss of information. So another way to build a perfect pattern is to just aggregate a bunch of things and lose information. Okay, let's do the same thing, but let's multiply the observations, right? You know you're going to get a log normal. So you start with these different things, you multiply random draws from those things, and you get a log normal. The two main drivers of pattern are the loss of information by aggregation and scale transformation. This is a scale transformation because multiplication is the sum of the logarithms. So this is just a log transformed Gaussian. Right? Everybody knows that. But the more general principle is the dissipation of information and scale transformation. We'll come back to that point later. So the common explanations are like this. You come up, you have a pattern, you make a microscale explanation, you fit the pattern in that way. You might be right, but maybe you're not. Maybe there's some other way to think about the problem. So what I want to do is give you a few examples of an alternative way in which we can generate this Hill equation pattern, which I think strikes me as more likely with regard to biological design. Let's think in terms of aggregation. Here we have a cascade of reactions. The input goes to an output. The output forms the input for the next reaction. The output goes to the next reaction. Here we have a cascade of 12 reactions. We're going to take Hill coefficients for each reaction drawn randomly from different distributions. Let's draw from this exponential distribution with a mean of 1.14. A Hill coefficient of 1.14 is visually indistinguishable from Michaelis-Menten kinetics. We just take random draws from this particular distribution with a mean of 1.14. We aggregate in a cascade, and we get a Hill coefficient of 1.7. The Hill coefficient multiplies through a cascade. You can get high Hill coefficients from components that have very low Hill coefficients. You can do the same thing by drawing from distribution with higher Hill coefficients, and you get higher Hill coefficients. The point is, you're getting aggregation is leading you to a significant Hill coefficient pattern. You can think of that as the dissipation of information, because in effect, this lower tail here is basically not transducing or transmitting the signal. And as you multiply that, you're getting a weaker and weaker transmission of low input signals. That's a deterministic model that shows the multiplication of the Hill coefficients. It doesn't tell you where you get a Hill coefficient of greater than 1 to start with, which was the original puzzle. Let's imagine a sloppy component. So we know that cells, individual cells can be quite sloppy, and that sometimes a cell, when it gets an input, just doesn't do anything. It doesn't react. That could be because you're getting stochastic fluctuations in the reactants that are involved. Sometimes cells are in quiescent states and don't seem to be doing very much as an individual cellular level. Let's just imagine this is the input signal, that's the output, and this is the input-output pattern, that for low inputs, the signal isn't transmitted occasionally. You're just getting a loss of transmission because the cell doesn't respond. So if you get a 30% chance of no response at a tiny input, and this failure to respond drops off to 0 very quickly. So you're just getting a little bit of failure for very low inputs. The cells are just a little bit sloppy. And now we do an aggregation of those in a cascade, where we have 2, 4, 8 of those in a cascade. What happens? Well, by the time you're up to 8, you've got a very strong Hill coefficient switch-like pattern. You aggregate a bunch of sloppy components that are just a little bit weak at transmitting low input signals. You put them in a cascade, and you're going to get a perfect Hill equation response. So if you're going to design a biological system, or imagine how biology is going to design something, is biology going to tend to design things by making very perfect, deterministic, microscale generative processes? Or is it going to slap together a bunch of sloppy components where the locus of design is really in the aggregate structure? So my view is that a lot of problems in regulatory control have to do with the modulation around these sort of attractors. Right? So biology can modulate the overall input-output relations by slapping together a bunch of sloppy components and taking advantage of the role of aggregation in giving relatively precise inputs and outputs. Maybe not so different from the first talk this morning, but trying to focus a bit more on the key aspects of what things are going on underneath. One last example, this one I'm kind of fascinated with, diffusion-limited reactions. If you take so this is a stochastic simulation, molecular simulation of a particular chemical reaction that I did myself. And if you do this reaction with high diffusion rate, you get Michaelis-Menten kinetics. If you look at diffusion limitation, however, you get a strong Hill equation response. Now, diffusion limitation is probably fairly common in cells because cells are often crowded. Sometimes the concentration of cells are low and things don't always diffuse so fast within cells. Now, why would diffusion-limited reactions give you a Hill equation response? I don't know exactly. That's the problem when you do a simulation. You have to figure out what's going on. But I think the reason why, one of the reasons why, is the following. Imagine that initially there's a uniform distribution of a non-active molecule and a uniform distribution of an incoming signal. So we have mass action, potentially ready to go. But imagine in a particular location that the signal molecule transforms a non-active molecule to an active state. Then in that little neighborhood, the concentration of the inactive molecules is now relatively lower. Now, it's the inactive molecules are basically measuring devices for the input, because that's what you're using the output, the transformation to the output or active form, as a measurement. You get a measurement in a local domain, and the system loses sensitivity because it no longer has the inactive molecule available to measure a new input molecule. If you have fast diffusion, that all spreads out, and you get perfect spatial uniformity gives you maximum sensitivity. So you lose sensitivity, you lose measurement capacity, you dissipate your ability to actually acquire information with each reaction in a diffusion-limited reaction scheme. So although there's a lot of discussion of diffusion-limited reactions in biochemistry, when I wrote to the person who wrote the program that's to study diffusion-limited reactions and asked him about the Hill equation, he said, oh, isn't that all known? That's just the whatever model you were going to describe, and there's nothing interesting biochemically. And I study diffusion-limited reactions. Why would you be interested in the Hill equation in diffusion-limited reactions? That's already well understood.
Could you say what the equations are that this is a simulation of?
No. I mean, I can find it in my paper, but I can't say right now because I don't remember.
What about the dimensions? Like many diffusions are not quite 3 dimensions in biology. Is that going to change the Hill coefficient? Or?
It all changes the Hill coefficient. The Hill coefficient is sensitive to pretty much every parameter that you can think of screwing around with. That's the point. I mean, there's, the details. We can, if anybody who's really interested in the details, we can discuss them. It's a molecular simulation. It's tedious and boring, but it's.
There's a reaction. There's an underlying reaction which is diffusion limited.
Yeah, there's a reaction.
And can you tell us what that reaction is?
I might have it. I might have it on a slide at the end. I'll see if it's there. So, I showed you 3 ways in which information might dissipate within reaction schemes. I'm not claiming that this is the way it happens, but the point is that it's actually fairly easy to build a Hill equation response from sloppy components because aggregation almost inevitably leads to that, or diffusion-limited reactions, or other ways in which information is sort of lost in these systems. So one of the key points then is dissipation of information is extremely powerful. And here's what I think is one of the great quotes in the history of science from the great mathematician Kolmogorov. In fact, all epistemological value of the theory of probability is based on this, that large-scale random phenomena and their collective action create strict non-random regularity. So in terms of biological design, take a lot of sloppy components, and if you stick them together, you get precision. So when we see precision, do we have to look for microscale generative mechanisms which are precise and perfect and difficult to maintain? Or does biology modulate using this principle, modulate sloppy components in terms of its overall architectural design to achieve relatively precise biological function? I'm inclined to think along these lines. Another quote from Jaynes, another favorite author: "We cannot understand what is happening until we learn to think of probability distributions in terms of their demonstrable information content." So I've been pursuing these ideas through a variety of articles. I want to make a key point in a second here, but I just want to mention the common patterns of nature. Is just this idea in general about these attractors. There's many different patterns. I mentioned at the beginning of the talk that biologists all know the Gaussian, but if you give them an exponential distribution, remember yesterday's talk about random chromosome breakage, you give them an exponential distribution and they're going to fit something to it and think they understand what's going on. The exponential distribution is an attractor. If you fix the average and you let everything else be as random as possible, any process that fixes the average and is pretty random is going to be an exponential distribution. So it's important to understand and recognize the common patterns. If you don't know the common patterns, you can't read what's going on in nature. So I've been developing this idea through a variety of articles: measurement, invariance, entropy, and probability. And this article, which I developed with my co-author Eric Smith, who is a really great mathematical physicist, and he really deserves the credit for this, but a simple derivation and classification of common probability distributions based on information symmetry and measurement scale. And I'm emphasizing this because one of the reasons why only the Gaussian tends to be recognized is that even in physics, and even in the sort of people who really know how to do this stuff, which isn't me, there still is not a really good understanding of what the real attractors are in terms of patterns. So we all know the Gaussian, we know the log normal, we know a couple of others. But there's actually a wide range of patterns which recur. For example, when I see a gamma distribution, which arises, for example, in the analysis of large data sets over and over and over again, many people think, oh, that's interesting, there's a nice pattern, I can explain what's going on there. And what I see is that the gamma is what you expect if you have a log linear scale and everything else is as random as possible. Let me develop that idea because that's the last point I want to make. Remember here, this is scale transformation with a Gaussian. You get a log normal. Everybody's got that. That's very common. But let's look at the Hill equation. So this literature on the Hill equation is vast. But I think that almost nobody, and I'm sure somebody has, almost nobody has ever said the following about the Hill equation. Let's just look at it. If the input is low, the denominator is essentially 0. We've got a power law. A power law is a log scale. This just says at low input, the scaling is logarithmic. At high input, you've got saturation, which is logarithmic scaling. So you've got log scale, and in between you get a linear zone. This is the simplest mathematical expression of log linear log scaling. Log linear log scaling, everybody does log linear log scaling. When you have small numbers and concentrations in chemistry, you write 10 to the minus 8, 10 to the minus 7, 10 to the minus 6. When you're in an intermediate zone, you write 1.1, 1.2, 1.3. When you're in a high zone, you say 10 to the 6, 10 to the 7, 10 to the 8th. We all use log linear log scaling. The Hill equation is just a mathematical expression of log linear log scaling. That is why it's so common. Log linear log scaling and a certain amount of dissipation of information, and you tend to end up on a log linear log scale. So when I say I see a gamma pattern probability distribution is what you get if you have a log linear scale, logarithmic at small magnitudes and linear at large magnitudes, and everything else is so random and dissipates so much that all you maintain are average values. At small scale, the average of the logarithm is the geometric mean, and at large values, it's linear, so you get the arithmetic mean. And the Gaussian is exactly that. It's the geometric mean and the log mean constrained, and everything else is random as they could be.
Okay.
So the common patterns, so more deeply, all of the common patterns, I believe, are actually fit into a hierarchy with regard to measurement scales. And that's what I did with my colleague Eric Smith, is that we've actually developed an underlying sense of the relationships between a variety of measurement scales. And it gets a little bit more complicated than just log linear, but there's a variety of scalings. And I think that you need to be able to read those patterns in order to read the patterns of nature and recognize them so that you don't explain what is in fact aggregation and dissipation of information by a complex explanation. Now, I'm not saying that the whole world is just based on aggregation, but what I'm saying is that this sets incredibly powerful contours for the nature of pattern, and that design in biology and computational architectures necessarily follow those contours and modulate those contours. And if you want to understand the computational architecture of natural systems, you have to understand what those common attractor patterns are and how biology modulates around those common attractors. So, to summarize, the first part of the talk was about the duality between robustness and decay. And if you think about that, what I was saying in the first part of the talk is that as you get systems that overlay on top of functional biological systems that protect biological systems from perturbations, homeostasis is what we call it in physiology, that what happens is that you tend to get a decay in the underlying components because those components are protected, and so you're going to use cheaper, lower performing components within the system. You get a decay, you'll get a tendency for sloppiness at the micro scale, but function at the larger scale. Okay? Then the second part of the talk, in essence, I turned it around and I said, when you see sharp patterns in biological systems, you should be thinking, maybe that's the outcome of the aggregation of sloppy components to give us precise function at the systems level. So the two different parts of the talk are actually sort of flip sides of the same coin, which is that you have to understand the relation between function at the system level and the sloppiness at the component level, and understand how evolutionary dynamics kind of makes a weird process that in the end gives us the architectures that we observe in relation to those two different points of view that are related to each other. So thank you very much.
Yeah, so I think it's an interesting point to what extent are the components sloppy? And I would say in the cases where people have actually measured as precisely as possible these input-output relations, it turns out that the components are in fact extremely precise.
Right.
So for example, in the cases, I gave the case of hemoglobin, but you can take, for example, even more physiological case in bacteria like chemotaxis, right? Every component of that system
I absolutely agree with the
is
point.
remarkably precise. So thinking of proteins as sloppy, I think is actually, it doesn't seem to be the case that proteins are sloppy.
I agree that that's, but here's the question. And I don't know the answer, so I'm not saying that I'm right. But here's the question. People see what they look for. A biochemist is very interested in isolating a system that can be studied in the laboratory in a precise way. And so people only study those biochemical reactions which have the characteristics which lead to what you say. They don't start by saying, I really want to understand biological design. And it turns out that this system's kind of sloppy, but I think that's really important for biological design, so I'm going to study it. That is not how biochemists think.
It's interesting just to find out, in which cases are the things more sloppy, in which cases are they not.
Well, that's the question.
Right.
Right, but I'm telling you why, in spite of the fact that that table was in that review article, and I think it's a beautiful table, and I don't think that any of those scientists are bad scientists. Mm-hmm. They've seen precision over and over and over again. But, yeah.
So you said you don't want to give an explanation that's too complicated, but similarly you don't want to give an explanation that's too simple either. So I give an example of the fashion in nature papers, with power laws and scale theory distributions. Everybody published a paper, something like the time it took for Einstein to respond to letters had this distribution, right?
Right.
So who gives a shit? What does it explain to understand that something fits a distribution? Okay, well, it could be something like if there's a general broad class of microscopic to macroscopic principles such as preferential attachment that cause that distribution, then that is it is, right? So I see methodologically what you're doing is proposing a broad class of principles that can construct distributions, and so, you understand deeply what constructs Gaussians, and now you understand deeply what constructs power laws. That's good. Is that roughly what you're doing?
I have no idea what you're about to say. I mean,
I've just said it.
That's one. Well, that's one.
I'll ask you a question. What do you want to do? I mean, is that roughly what you want to do, to define a broad class of mechanisms that constructs Hill function-like things?
No.
No?
No, absolutely not.
What do you want to say then?
I want to understand biological design. I want to look at an organism, see how it's put together, and understand how it's come to be that way. I believe that the current approach, which was emphasized very strongly by our friend back here, about the way biochemists view the world, although I think that their contribution is immense, I think it's only a tiny fraction of what goes on in biological design. I think that a lot of biological design is actually based on the aggregation of sloppy components.
So that's right. So you're identifying a broad class of principles which constructs that distribution, which is what Strogatz and other people do with their distributions, right?
I don't know how, I don't know. You're saying something that you think I'm saying, but I don't hear it.
So just one comment and then I guess maybe the other part's a question. So my impression was about hill functions, it is the general shape that you see all over the place, but exactly that it being x to the k here and 1 minus x to the k here, you know, you do T-thel fits, it doesn't usually look like that. There are some cases it does. So it's a question, which is one mainly trying to explain these steep curves in the middle for this highly sensitive behavior. I guess the question is, so there's a very simple general mechanism that gives you things that look like this, as far as the general features, which can be, you can make sharper and less sharp by changing parameter. And that's just to observe that one of the very generic things that come out of all kinds of things is actual bistability. Not where it looks like this, but where it's actually bistable. There's a stable thing here and a stable thing there. And you can change something and it'll flip between them, but it doesn't flip without changing. Okay. If you take that, then you add sloppiness. So you add stochasticity, and the stochasticity on faster on the time scales than you're looking at, then you can get something with arbitrary steep curve in the middle by just how much stochasticity you have, right? And so it's a very general thing because we know, double pairs of fixed points are very general, and the stochasticity in there as well. I mean, does that count as an explanation of the kind you're looking for, or is that?
No, all common patterns are common for one reason, because there's a huge number of mechanisms that generate that outcome. So if you look and you think, I've got a way of explaining that, I'm sure you do. I'm sure everybody does.
I have no biochemistry in there. I just say absolutely generically, you get bistability, and then you would absolutely generically get fluctuations, and therefore absolutely generically you get those kind of curves.
If that works for you, that's fine. I'm not sure what your question is, though.
But why is that not? Why is that not something of the kind you're looking for?
I'm looking for understanding organismal design, and I'm looking for how organisms are put together. And I think that organisms are put together by taking advantage of the fact that there are powerful attractors that set natural contours of pattern.
If dynamical systems that have bistable is a natural attractor in any reasonable sense of attractor.
Okay, then it works.
Well, we should probably have lunch and continue the discussion if
Thank you.
doesn't get very good. We should come back at 1:30. Thank you.